Okay, well, now we are in Chapter 6. Moving into this next part of the course, we are going to be talking about more neurotransmitters. So in a previous chapter, we really focused on the nature of the synapse and how synaptic transmission worked. And of course, that involved some discussion of neurotransmitter systems. And when we now move into this particular topic of what we're going to call, let's say, psychopharmacology, this is a field that involves understanding the psychological effects of pharmacological influences on the brain. Specifically, of course, then we're talking about drugs that influence the brain, psychoactive medication. One thing we want to review, this was partly covered in a previous chapter. Sorry, there's a misspelling. I just see that on the title there, neurotransmitter systems. And of course, there's fairly more than these, but these are four of the more widely understood and studied and primary neurotransmitter systems in both the central and peripheral nervous system. So especially say the cholinergic system that uses choline or acetylcholine works in both the central and peripheral nervous system. So in the peripheral nervous system, we know the important role that it plays as part of the autonomic nervous system. And so, of course, it affects the internal organs, and it plays an important role in muscle contraction. But in the brain, it plays important roles in learning and memory. So anytime we see changes in neuron activity in the brain as a result of learning and encoding of memory, we know that choline plays an important role for that. There's research, for example, this is a little beyond the topic that we're going to get into in more detail, so I'll just mention it right now, that if you boost choline activity in the brain, that tends to be good for memory formation. And there are various drugs that inhibit choline in the brain. These are called anticholinergic drugs, right? Examples of this tend to be things like antihistamines, right? Benadryl and similar kinds of medications. These are all anticholinergics, and they can, depending on, of course, the length and duration of their use, lead to some memory impairments. So maybe I think especially the case in older adults. So older adults generally are not recommended to be taking anticholinergic for a long period of time because it could have an impact on memory function. Conversely, of course, some of the drugs like Aricept that are used to treat Alzheimer's disease are cholinergic drugs, right? They boost choline in the brain, right? And so I wanted to mention that real quick because that's a kind of a relevant point to understand what these drugs do in terms of neurotransmitter effects. And it's not a detail I had later in the slide, so I just wanted to mention that. Then, of course, there's dopamine in the brain, and so that's referred to as the dopaminergic system. We know that its role in the brainstem and cerebellum is related to the control of movement, but projections that go into the frontal lobe of the brain seem to be involved in processing information about reward and feelings of achievement, accomplishment that would generally be considered a pleasurable feeling, come from the so-called dopamine reward pathway that's been popularly discussed for a few decades now. Serotonin is part of the serotonergic system, and its derivatives like melatonin are all relevant to our circadian rhythms, the sleeping and waking cycle, general arousal levels in the brain. The noradrenergic system is important for learning and also emotional states. This is why many antidepressants are things that affect the noradrenergic system, right? There's, of course, famously the concept of the antidepressants influencing serotonin levels in the brain, and that's still, of course, a common approach to treating depression, but it's worth noting that recent papers have been coming out in the neuroscience field showing that the so-called serotonin hypothesis of depression does not have the evidentiary support that we once thought it did. So this was a meta-analysis. And if you don't know, a meta-analysis is when you do a review paper, so these are useful to do sometimes in any science, is to review a given topic. So you can sort of describe and summarize all of the studies that have been done on a given topic to look for trends and say, okay, what do we really know about this topic, and what do all these studies show, and what do they don't show, and things like that. But a meta-analysis is when you take that a step further and you start pulling data from all of these papers and you combine all of that data into one big sort of general analysis to say that, okay, one study found positive effects, but another one negative. And so if we add all of that up, this is a simplification, of course, what's the sum total of it all? And in this case, the sum total was not very much. So seems to suggest that serotonin might not be so significant for understanding depression as we once thought. But nevertheless, the serotonin-based medications are still used and may still have some effectiveness for reasons that we don't totally understand at this point. So, of course, that's the theme of this topic here, is we want to understand the nature of drugs and how they affect the brain, and especially working at the level of the synapse. So these are all called psychoactive drugs. And related to that is a question of why do people develop dependency or addiction-related issues when it comes to these drugs. So very, very brief overview at this point. Some drugs work because they mimic a neurotransmitter. And what that essentially means is that the molecule of this drug has a shape that is either identical or at least very, very similar to a neurotransmitter. And as we've learned previously, neurotransmitters are sort of like a key, and that unlocks a door in the postsynaptic membrane. And in this particular case, of course, a drug that works as that mimics that neurotransmitter by having the same molecular shape would activate those same receptors, unlocking them and triggering the appropriate response in the postsynaptic cell, whether that's an excitatory, inhibitory, metabotropic response. It would influence those postsynaptic membranes in the appropriate way. In other cases, drugs don't necessarily influence, or I should say mimic, a neurotransmitter, but they may influence other aspects of the synapse, right? So drugs can do things such as change a metabolic process in a neuron that could either increase or decrease the amount of neurotransmitter that it produces. But if you know about this, we're just talking about the serotonin drugs. And so the classic antidepressants are called SSRIs. That's called selective serotonin reuptake inhibitors. And in a previous chapter, again, we talked about how once synaptic transmission is complete, one of the things we need to do is reuptake the neurotransmitter into the axon terminal so that it can be recycled. And SSRIs interfere with this process, and that allows serotonin to remain in the synapse a little bit longer, and therefore increasing its chances of activating receptors on the postsynaptic membrane. Let's first talk about addiction and dependency-related issues. Now, there's a long history of trying to understand the nature of how certain drugs might affect the peripheral nervous system, right? And so the balance between the sympathetic and parasympathetic nervous system can be shifted in one direction as a result of certain medications and drugs. And that once the body and the autonomic nervous system gets used to that, that becomes kind of like the new normal for the autonomic nervous system. And then when you stop taking the medication, that disrupts that, and that causes a variety of autonomic symptoms that are oftentimes rather unpleasant. This is what we typically call withdrawal symptoms, right? At least at the physical level. But a lot of times, drugs that are addictive also result in what people report as more of a psychological withdrawal. That is a feeling of need that they have to take that drug. And a lot of times the drugs that could lead to this kind of addiction-related feeling don't have the same effect on the peripheral nervous system. And on top of that, it's worth pointing out that a lot of times people become addicted to behaviors rather than simply just a drug. So we know that there are such things as sex addiction and gambling addiction and internet gaming disorder and shopping addiction and things of this nature. And we wonder, is this the same underlying neural mechanism involved in those kinds of addictive behaviors that might be involved in addictive drugs? And what we see is that, yes, there is potentially an underlying component here, because all drugs that are associated with problematic use all have an effect on the dopaminergic system in the brain, that infamous reward pathway that I had mentioned earlier. And like I said, this has been known for a long, long time. This goes back to 1954, the Olds and Milner experiment. What they had done is they had inserted microelectrodes into the brains of rats so that any activation of this electrode would activate the nucleus accumbens. It would stimulate neurons in the nucleus accumbens, and that's part of that dopamine reward pathway that goes from the midbrain through the nucleus accumbens and further on into the frontal lobe of the brain. I have a map of that, so let me just take a look. Let's take a look at that real quick on this picture. So we see the pathway there in blue taking us from the midbrain through the nucleus accumbens, which is a part of the frontal lobe, and then from there, pathways that spread out through other parts of the prefrontal cortex, right? And so let's come back here. So now by inserting in a rat brain, that was a human brain, right? But in a rat brain, if we insert these microelectrodes there, then any activation of that electrode stimulates this reward pathway. Of course, it generates these feelings of pleasure. What they found is that they were able to attach this, the circuit controlling this electrode to a lever inside a Skinner box. And when they placed these rats inside the Skinner box or operant chamber, the rats could press the lever, and what they would discover is that immediately upon pressing the lever, you get that so-called dopamine hit, right? It feels very pleasurable. And here you see the results is that these rats would end up doing this up to 2,000 times per hour, right? They really were, in a way, you could say, addicted to lever pressing, right? And so it seems to be the case that there's nothing inherently addictive about pressing levers. You don't see rats in other kinds of situations become addicted to lever pressing, right? But only when it's set up to activate the nucleus accumbens that we see this kind of result. And that seems to raise these issues that, is there something about the stimulation of this particular pathway in the brain that is involved in addiction? And as I said, of course, so many of the drugs that people can become addicted to activate this pathway in the brain. And likewise, there seems to be a parallel to a lot of these behavioral issues that sometimes people develop dependency issues around, right? So let's get back to the effects that the drugs have in the brain. So sometimes, one thing to keep in mind is that this language here applies to whether or not the drug is mimicking a neurotransmitter or whether the drug is affecting some other stage of the synapse. But what they're going to do here is that they're either going to increase the effect of a given neurotransmitter, and that's what I kind of mean by exciting activity at the synapse. They're increasing the neurotransmitter action at the synapse. Or they are inhibiting neurotransmitter action, right? And they're decreasing the effect the neurotransmitter has within the synapse. So any drug that does that seems to block the neurotransmitter in some way is considered to be an antagonist of that system. So you can have, like I said, with those anticholinergic medications that can block the effects of choline in the brain, those would be considered choline antagonists, right? Meanwhile, of course, any drug that increases the effect of a neurotransmitter in the brain is an agonist of that neurotransmitter. So I had mentioned Aricept as something that boosts choline activity as a treatment for Alzheimer's disease, and so that's a choline agonist, right? We have a few other things that we can say here, just a few key terms here. So when we talk about a molecule of a drug having a similar shape to a neurotransmitter, and that means it could bind to those postsynaptic receptors on the dendrites of a neuron and activate those receptors. But this depends on just how similar that molecule is, because sometimes you can get away with a little wiggle room here. So we can talk about the degree of affinity a drug has for a particular receptor on a postsynaptic membrane. So high affinity would mean that the drug has a very, very similar molecular shape to whatever neurotransmitter it's mimicking. And then a weak affinity means that it's not quite so similar, so that it does not particularly bind well to these receptors, and it might occasionally create an effect and open up a few receptors, but maybe not consistently. And that then relates to what we'll call the efficacy of the drug. So efficacy here is the degree to which this drug can actually effectively activate these postsynaptic receptors and trigger whatever response on the postsynaptic neuron that we would expect. Again, whether it's an ionotropic or metabotropic response. And so we can see in this case that it is possible that sometimes drugs might have a high affinity and they might stick to those receptors, but they don't always unlock the receptors. So that's a curious result. So in this case, where a drug might lock onto certain receptor sites, but not fully unlock the ion channels that are controlled by those receptors, this actually might work in a way that is as an antagonist drug, right? Because in this case, the idea is that the drug is binding to the receptors with a high affinity, but because it's not unlocking the ion channels, the postsynaptic membrane is not responding. And meanwhile, anytime the presynaptic axon terminal is releasing the neurotransmitter, it doesn't have anything it can do because the receptors are all being blocked by that drug with the high affinity, right? And so that would be an example of high affinity, low efficacy. Let's have a look, though, at different kinds of drugs. Here we have a variety of different stimulant drugs. Some may be a little bit more of a hybrid, like I'm looking at MDMA here in the middle of the slide. But what these all have in common and what makes them all to some degree stimulants is the effect that they have on dopamine, right? Typically, stimulants are categorized as pretty strong dopamine agonists, right? So amphetamine in any form, like methamphetamine, for example, is a strong dopamine agonist, right? And in this case, we see what it does is it increases the release of dopamine from the axon, right? So I said that earlier, that in some cases, what the drug does is it increases how much of a neurotransmitter is produced and released by an axon terminal. And that's exactly what many amphetamines do, is they just really flood the synapse with excess dopamine. Cocaine is also a dopamine agonist, as well as being a norepinephrine agonist and a serotonin agonist, right? So in this case, what's happening is that it blocks reuptake. So it's a reuptake inhibitor. This means that dopamine stays in the synapse, norepinephrine and also serotonin stay in the synapse. Methylphenidate is brand name Ritalin here, is one of the many ADHD medications. The other ones, Adderall, are actually a variation of amphetamine. So you can see the effect is a little bit different. So Adderall is going to work more as an amphetamine, and it may increase dopamine levels. Methylphenidate, on the other hand, is a reuptake inhibitor for dopamine. So again, it's an agonist, but it works with a slightly different mechanism. And it also does so in kind of a slower, more gradual way. So in this case, it works a little bit differently than amphetamine and cocaine do, especially comparing that to cocaine here, where cocaine has a much of a really strong blocking of the reuptake effect. Methylphenidate's effects are a lot milder, and for that reason. Now, MDMA, as I had kind of hinted at before, also known as ecstasy, is a difficult one to categorize because it is known to create hallucinogenic effects, and this is associated with the increase in serotonin that it triggers, right? And so drugs that increase serotonin can have certain hallucinogenic properties. It's a common thing with most hallucinogens, but MDMA also creates a strong boost in dopamine as well. And that seems to be associated with some of the pleasurable feelings that are commonly reported with the drug, hence why it is referred to as ecstasy after all. Nicotine is it mimics acetylcholine, right? So it has an effect on certain kinds of acetylcholine receptors. There are actually at least two key acetylcholine receptors. There's the nicotinic receptor, and hence the drug nicotine will activate that. There's also something called the muscarinic acetylcholine receptor, which we're not going to really talk too much about here. But the idea is that these nicotinic receptors are found in the central nervous system, They're also found actually right here, the nerve muscle junction. So when we talk about a motor neuron in the spine that sends an axon out to a muscle and synapses onto the muscle to trigger muscle contraction, those are nicotinic receptors there. And also in the nucleus accumbens, of course. So we see, so of course, this is a different neurotransmitter. It's not dopamine, it's a cholinergic pathway, but there are cholinergic neurons involved in the reward pathway of the nucleus accumbens as well, as it turns out. All right. In fact, this is really true. We don't want to oversimplify the matter here and suggest that these different parts of the brain use only one neurotransmitter, because no, they use lots of different neurotransmitters and for different purposes and different effects. Speaking of drugs that have a similar molecular shape to a neurotransmitter, opiates, right? So these are things derived from opium, and this includes, listed at the bottom, morphine, heroin, methadone, various other drugs. They are very, very similar to what are called endorphins. And endorphins are hormones / neurotransmitters. And one of the things that they do is they create an effect on these two neurotransmitters of GABA and dopamine. We haven't really said much about GABA, so there's a whole separate neurotransmitter called the GABAergic system. And these in part play a role in some of the pain pathways in the brain. And we have a chapter coming up that's going to talk more about how touch information from the body is transmitted to the brain, and especially what's known as the nociceptive pathway, the pain pathway. But these endorphins are neurotransmitters that are important in activating the kinds of effects that GABA and dopamine have on this pain pathway. And because these drugs influence that or mimic that effect, you get pain relief from it. But they also have effects on for example, the autonomic nervous system. And this is why, for example, you will see that large doses of these result in slow heart rate and respiratory suppression. And so an overdose of these can be fatal for that reason, because it slows down the heart rate to the point blood pressure drops very dramatically to dangerous levels. And respiration drops to very low dangerous levels. And so low oxygen, low blood pressure, these are all very dangerous things. So this is why one of how these opiates are not just affecting the central nervous system and creating some of the pleasurable effects associated with dopamine, as we see here, but also in this case, suppressing a lot of the parasympathetic system. Or I should say magnifying the parasympathetic response, right, by actually slowing down. That's what the rest and digest parasympathetic does. Now, I've already noted that hallucinogens tend to be associated with serotonergic agonists, right? And so I have that listed here. So the definition is just basically anything that has an alteration over conscious experience and perception. So visual and auditory hallucinations are common with many hallucinogens such as LSD and psilocybin. Add in some tactile hallucinations associated with MDMA, but serotonin is a common to a lot of these hallucinogenic drugs. One of the things that we see is that we know that the visual part of the brain and the occipital lobe, again, another chapter coming up, uses serotonin as one of its core neurotransmitters. And there are various different kinds of serotonergic receptors in that part of the brain. And so the fact that we're stimulating some of those receptors, this might explain why you end up with some of these visual experiences. Now, for some of the other stuff, other kinds of modalities, that may be less clear. For marijuana, the key psychoactive ingredient is tetrahydrocannabinol, or THC. And we know that there are receptors in the brain for this as well. So these are called the cannabinoid receptors. There are many different cannabinoid receptors, but two of the key ones that have been identified are CB1 and CB2. We find these all throughout the brain, but we find a lot of them in sort of that brainstem stuff. So we find them in the cerebellum, the basal ganglia, which is part of, both of those two are part of motor control. And the hippocampus is important for encoding long-term memories, right? We see some interesting memory-related effects here, right? So it's known that memory impairment occurs with THC use. There can be some alteration of sensory experience. So that, again, that could be related to either a serotonin effect that is not totally understood here, or maybe something about these cannabinoid receptors that affects that as well. And the subjective loss of time, that one is maybe tricky to explain, but it could also be the case here that if the hippocampus is being impaired and it's struggling to embed long-term memories, it might be that our sense of time is associated with sort of this continuous flow of memory encoding. And if that's disrupted, then our sense of time could also be disrupted. And by the way, why do we have cannabinoid receptors in the brain in the first place? What are the internal neurotransmitters that activate those? It's not THC, it's other things, anandamide and 2-AG. These are the internal, what are called endogenous compounds or neurotransmitters that THC mimics, right? Likewise, there's another compound commonly found in marijuana called cannabidiol, or CBD, and different versions of this may or may not have very noticeable psychoactive effects, but of course there are commonly used medical versions of CBD now that are available very, very commonly. That activate the CB1 and CB2, but less so than THC does. And instead, they activate a variety of other of these related cannabinoid receptors throughout the brain. And since they don't necessarily produce psychoactive effects, so we wonder, what are they actually doing? And that's harder to study because obviously research on these drugs has been limited over the last several decades because of their legality issues. But there's a lot of interest in the medical community about studying the anti-inflammatory effects of CBD. And this seems to imply that the effects here might be related to the same receptors that we find for the stress hormones. And so stress hormones cause inflammation in the body. And therefore, if CBD disrupts the effects of the stress hormones and blocking the receptors for those, then maybe that could explain these findings. But of course, there's a lot to be found there. Well, that's a short and sweet chapter. And that kind of finishes up our discussion of neurotransmitters and the synapse. And we will be moving on to the next chapter next.